Hello and greetings from LIU. Here's a picture of our campus in downtown Brooklyn, a nice little oasis in the city. My name is Tim Leslie, and I've been a professor in the Department of Biology for 12 years now. And my area of expertise is in entomology, or the study of insects. And so my lab is primarily interested in studying how we can conserve beneficial insects that provide us with important ecosystem services. For example, the bees you see in this picture here provide us with important pollination services in the agricultural sector for many different types of crops. From a teaching standpoint, you can find me teaching anything from general biology to sustainability science to even advanced ecology. I'm a big fan of active learning, experiential learning. So here you can see one of my classes visiting the largest rooftop farm in the world, which happens to be just a few blocks away from campus. I'm also the founder and advisor for the Campus Outdoors Club. And so this club allows students to get out in nature, go for hikes. We also do a lot of volunteer work in the local community as well. I call my lab the Insect Ecology Lab, and I'm lucky to have a lot of students involved in research, ranging from local high school students to even graduate students. Here's one of my former graduate students, Mohammed, doing some field sampling in a community garden in downtown Brooklyn. Here are some undergrads, Nasia and Crystal, doing some microscope work and crunching numbers. I also encourage all of my students, like you see Harjeet and Alicia here, to present at professional conferences as well. Another important aspect of my lab is to do outreach with the public. And so here I am at an environmental science fair with another student where we had a table and we're interacting with anybody who had any questions about our research program. So today I thought I'd talk about just one of the numerous studies going on in my lab, and this one dealt with honeybees. So honeybees are arguably the most important insect in the world. They're definitely our most valued pollinator, and the reason they're so highly valued is that they exist in these large colonies that are easily transported on an as-needed basis to different cropping regions. And so it's believed that about a third of our diet comes from honeybee pollinated crops, and that includes a lot of the wonderful fruits and vegetables that we enjoy every day. Agricultural economists estimate that about $20 billion worth of produce is attributable to honeybee pollination on an annual basis, actually, in the US alone. So thinking of the importance of honeybees, it was of great concern when we noticed that their colonies started to decline at alarming rates starting about a decade ago. And so as a result, there was a concerted effort among the research community to figure out what was going on with these honeybee declines and what was causing it. And these directives for research went as high up as even the White House, which established a pollinator health task force in 2015. And since then, there have been numerous avenues of research, numerous hypotheses put forth as far as what's contributing to these losses. And one major area of research is the relationship between pesticides and honeybee declines. And so there have been a lot of lab-based toxicity trials. However, there's still a need for understanding how honeybees interact with pesticides in a real-world setting. So one such study looked at honeybee colonies from all across North America and collected pollen and wax samples and looked at pesticide residues. And they found an alarmingly high number of pesticides and relatively high number of concentrations too of these pesticides as well. Our lab noticed that despite these types of studies, there were very few being done in urban locations. And keep in mind that there's been a rise in urban agriculture, and even New York City, for example, now has legal beekeeping. So we designed a local study to look at honeybees and pesticide exposure in the New York City region. And so we came up with two straightforward questions. One was, are urban bees exposed to pesticides at all? And if so, can we detect a pattern in pesticide exposure across what we call an urban gradient? So from the center of the city, area of high urbanization, out to the suburban and even rural areas outside the city at areas of low urbanization. And here you can see an example of an urban apiary on a rooftop in Brooklyn, New York. I just want to walk through then the steps of the research process so you can sort of envision what we did. The first step was to establish and describe our field sites. Remember that we wanted to look across an urban gradient. So we chose sites not only in New York, like this one in Manhattan, Brooklyn, and Queens, but also in the surrounding regions like these in Long Island, and these in Westchester that went from even suburban to more rural locations. We then wanted to characterize the landscape around each of these sites. Um, and so we used a specialized mapping software. You can see this map here. These different colors correspond to different land use types. So as you might guess, the deep red is impervious surface, like streets and buildings. And then as you move outside the city, the greens and yellows are like forests, meadows, agricultural areas. And so for each site, we measured urbanization by looking at the population density using census data. And then we looked at the amount of different land use types, such as agricultural area. And we specifically did this at a 6-kilometer scale because that's the foraging range of honeybees. So keep in mind, we want to tie this all back into these honeybees that are leaving the hive, going out to collect pollen, getting nectar, and then bringing that back to the hive, potentially exposing, right, the other bees to these pesticides. We then did our field sampling. And so we decided to focus exclusively on pollen coming into the hive. And so the beekeepers we worked with agreed to put these external traps, and it forced the bees to go through a special entrance that allowed us to extract the pollen off their hind legs into these collection bins. And here's one of my students, Andrew, with one of these pollen samples. And remember, our sites were from rural areas like this all the way to urban areas. This one was on a rooftop of a skyscraper right in Times Square in Midtown Manhattan. So very interesting field sites in this study. We then wanted to know what type of pesticide residues were on these pollen samples. So we worked in coordination with a USDA diagnostics lab that could analyze the pesticide residues on these pollen samples at a very high resolution, at parts per billion, for hundreds of different types of pesticides. Now, keep in mind, anytime we get a detection, these different pesticides have different toxicity levels as established by the EPA to honeybees. So not all pesticides are the same. So for each sample, what we did is we calculated what we called a hazard quotient, which is a way to characterize the potential threat to the honeybees from that group of pesticides found on any given sample. So you're reducing all that complexity into a single value for each sample. What did we find then when we summarized our results? So one thing that jumped out at us is that we had very few pesticide detections. In fact, an average of about 1 per sample. And within the New York City boundaries, all these pesticides were found at trace or low levels. However, when we started moving outside the city, we started finding higher values for different pesticides. So these are color-coded in this table here. As you go from left to right, this is more urban to less urban, to more suburban and rural. And you can see the lighter colors are trace or low amounts. The darker colors are higher toxicity or higher concentrations. And so you can see sort of a pattern emerging here. And then remember that hazard quotient looks at the potential threat to bees. Only one site had a very high value. It was greater than 5,000, which is approaching potential toxicity to the bees that might be exposed to that pesticide residue. As far as the research process, this is probably one of my favorite parts when we actually do the statistical analysis. We build the models. So remember, our second question is we wanted to know, was there a pattern across the urban gradient between pesticide exposure and urbanization level? And so we were able to find a significant relationship between these variables. For example, when we looked at population size, as population size increases, we got a decrease in the hazard quotient or pesticide levels. So that was as expected, right? As you go towards the center city, there's fewer detections of pesticides. The second graph is showing the same thing, just with a different variable. Here we're looking at the amount of agricultural land, right? And so as you have more agricultural land surrounding a site, you see the hazard quotient actually increases. So these two graphs are showing the same thing in a slightly different manner, but we were able to find a significant relationship between these variables. So what are some of our key takeaways? So the first one is just that the experiment worked, right? That we can actually use honeybees to monitor conditions in the surrounding environment, in this case, pesticide levels. And even at one site, we found levels that were potentially toxic to those bees. However, our biggest takeaway was that urban areas actually may act as a refuge for honeybees from pesticides, unlike what we see in rural locations. And so overall, we did find very few. And so this is some encouraging news for those of us that live in cities, for example. And lastly, we were able to find significant relationships between urbanization and pesticide levels, those models I showed. And so those could be very helpful in future risk assessment efforts as scientists continue to try to understand what causes colony declines in different regions throughout the country. I'd just like to finish then by saying thank you for listening. Hopefully you found that interesting, and I hope to see you in the classroom or in the lab in the near future. Okay, take care.